DNA, which can come and may go. For example, during cell divisions, when chromosomes are condensed, this would be retracted. That's the reason why you don't see nucleolus in dividing cells. When chromosomes are retracted, when chromatins are retracted for packing and forming chromosomes, you don't see nucleolus. And this is what the pars amorpha of the nucleus looks like. So this means that this is what I've shown you. This is part of the DNA, and look at this region is transcribing. So this region is the pars amorpha found within the nucleolus. This one is just showing only on one chromatin. Think of you have 10 of this type. This could come at one location to give you one nucleolus, or multiple to a maximum of 10, which does not even happen, to give you such multiple nucleolus. Then the second region is this type of things. This type of things and this one. This one is a recently transcribed ribosomal RNA. So it is a primary transcript, which is fibrillar in shape. It is small segments of primary transcripts of RNA. In addition to this, we also need to have imported proteins, ribosomal proteins from cytoplasm. Site of protein synthesis is not within the nucleus. It is in the cytoplasm. So these ribosomal proteins are synthesized in the cytoplasm and then should get through the pores, look at this, should come into the nucleolus. So these are the ribosomal proteins plus recently transcribed ribosomal RNA would appear like this one, like this one, like this one. We call it pars fibrosa. And the third one is when these proteins and this ribosomal RNA get assembled together to give you a recently assembled ribosomes. This appear as granular type of bodies, name them pars granulosa. So in the nucleolus, you can distinguish such three structures, which are just nothing on the process of making and assembling ribosomes. These are nucleolus, which may come as a single or multiple. When cells are engaged in production of large amount of proteins, the nucleolus becomes multiple in dividing cells. When cells are resting, they would appear as smaller in number as well. And then the background is giving us nucleoplasm. It is an amorphous ground substance like media. It is made by structures other than the chromatins and the nucleolus. It is composed of interchromatin granules, perichromatin granules, as well as nuclear matrix. So other required substance and structures found in the nucleolus, which are categorized under any one. So any one of this is what we name as nucleoplasm. So that's what I can tell you about the nucleus with regard to its makeup and structure. And then finally, briefly, I have to say a few things about cell cycle, because cells are divided. They have lifespan. They have a time of formation, growth, activities, if needed, division. So that's their cycle. It is not like individual organism. An individual organism, of course, made by cells and tissues, is formed as a small, and it grows and then ages and then dies. Is this not the case in cells. When cells are formed, they are already almost size of the parent cells. Of course, a little bit smaller, they grow. And then they get into their activities. And when they want, they divide and then continue. So they have a cycle. Of course, they have also time when they die, but they die altogether when the organism dies. Otherwise, as far as the person is surviving, we say that the cells are in a cycle. This cycle could be divided into two major phases in a somatic cell. One is interphase, the other one is mitosis. These are the two phases in a somatic cell. But in gametes, during gametogenesis, in addition to this, we also have an additional cell division, which is meiosis. This will be covered after a break by my colleague, Dr. Solomon. So you will see that. I'm only to show you about cell cycle now. This type of cell cycle for the cell is triggered by events of external to the cell, by factors coming from the outside. These factors may be multiple. One of the important factor is hormonal regulation. Regulation by, for example, human growth hormone. When human growth hormone is produced, cells are able to divide. They would be stimulated and cells are dividing. Lack of human growth hormone could stop cells dividing, so it inhibits, so cells are not dividing. It leads to just a small-sized individual. But therefore, this is one example. But there are also other reasons why cells are dividing. Yesterday, for example, I told you some cells are resting. Like I gave you an example of our liver. Liver normally reaches to its maximum number of cells and cells stop dividing. But if needed, for example, when cells are damaged and some are dying, to replace them, cells begin to divide. So you see, cell death can also initiate cell division. Size of the cell. When cells are excessively large in relation to the normal size of the cell, cells begin to divide. So there are external factors which trigger cell division or inhibit cell division. The actual cell division is going to take place by activities within the cell. So there are series of activities inside. There are stimulation, there are communication of messengers within the cells, leading ultimately for cells to divide. And this have phases, one stage getting into the next stage. Before it gets into the next stage, the activities which have to be taken place in that previous stage should be checked. We call this one as checkpoints. We have specifically in cell cycle 3 important checkpoints, 2 of which I'm going to show you soon. And this are what are happening and for cells to divide and the rest, giving us what is known as in somatic cell cycle as interphase and mitosis. Mitosis is a moment of cell division. It is a brief one. It doesn't last long. It is about 30 minutes to about 3 hours in general. It could be more or less, but it is a brief one. Whereas interphase is the longest one. And previously, interphase was thought to be a period of rest, while mitosis is a period of activity, the activity being cell division. But for cells to divide, I'm telling you, there should be some important things happening within the cell. So this means that interphase is not a period of inactivity. It is even much more active phases. And it is also a period when cells are growing. It is a period when cells are also accumulating, synthesizing and accumulating important substances needed for later cell division. And as I told you, it is the longest period, longer period compared with the mitosis. It is therefore could be represented in this chart, in this diagram rather. Here is the short period, the short phase of mitosis, but the longer one is interphase, which could be divided into 3 phases. The first one is named as gap 1 phase. The second one is the synthesis phase, when DNA is actually replicating, and the second one is named as gap 2 phase. So in interphase, we have a G1 phase, S phase, G2 phase to be looked at. G1 phase is the longest. It is the longest and the most variable phase. I told you cells, with regard to their cell cycle, could be divided into 3. I told you this yesterday. Now, for the last time, let me revise what I said yesterday. The first type is labile cell. Group of cells are named as labile. Labiles are short-living, so their cycle is very quick. Within a week, you can have cells dividing. Within a week, you can have cells are getting replaced. You have this type of cells in regions that are subjected to stressful conditions, frictions. Like, for example, while you are chewing your food, while you are swallowing bolus, the lining epithelial cells are sloughed off. They are easily get damaged, so they need to be replaced quickly. Those covering our skin are easily get damaged, so they have to be replaced. New ones are forming from the bottom. They are moving up, getting aged, and be removed by cells coming from the underside. This type of cells may be found in different layers, or you can also have a single cell layer being replaced quickly, like those found in the lower part of our gastrointestinal system, in the stomach, lining the intestine. They are only single cells, but most of these are to be replaced very quickly, because this type of cells are labile or short-living cells. So their cycle is shortest. So G1 is also short. But for those which are stable cells, like, I repeat again my good example, liver cells are stable. Once they reach to the maximum number, they no longer divide. They stop their division. So their G1 phase is longer, and they carry on their activities. If needed, they can come back to divide. So we call this type of cells quiescent cells, or the are you with me? Are you with me? They are resting cells. Whereas other cells, okay, thank you. Other cells, such as the neurons, are no more dividing. We call them permanent cells. So cells have different lifespan. And because of this, the G1 phase is very variable. Some noise is coming, I don't know from where. Could we mute? Okay, I can mute myself. Please mute yourself, otherwise noise is coming. Maybe it is from the administrator place. Okay, thank you very much. Now the noise has gone off. So here we have the G1 phase to be variable in it is period. And it is during this period that required RNA and proteins are synthesized. It is during this period that cells are able to grow to their parent sizes. It is during this period that the duplication of centrioles begin to take place. And once all these are formed, the integrity of the DNA is also assessed for the next period, when it needs to be replicated, when it needs to be duplicated. So if it is mutated, we are going to have this to be duplicated and creates this type of abnormal type of daughter cells. So it has to be assessed, it has to be checked. In fact, the assessment of the integrity of the DNA is a checkpoint. If it is just the type of required DNA, the checkpoint would inform the cell to proceed into the next phase. If the assessment makes the thing to be not adequate, the cell will not proceed into the next phase. So this is a checkpoint. For proceeding into the next phase, there should be some enzymatic activities taking place within the cell. So this is an important thing to know, and we call it a checkpoint. If the cell is not required for any reason, maybe the reason may be the cell happened to be not to replace itself because the number of cells may be adequate, like in permanent, like in stable cells. So it will not, the checkpoint will not allow it to cross into the next phase. So it remains in a condition of non-dividing cells. In this case, this phase, this elongated, otherwise elongated G1 phase would be no more G1. Instead, we call it G0 phase. We call it G0 phase. So in here, the cells are remained without crossing into the next phase. So the cells are out of the cycle. We name this one G0 or G0 phases. It is in this phase that stable cells and the permanent cells are located. When needed for quiescent or stable cells, other type of proteins would trigger the cell. So it comes to G1 phase and then crosses to the next phase. In permanent cells, there is no protein to trigger it to come back to the cycle, so it remains there, and we call this as permanent cell. So you see, there is a side branch of G1 phase, which is G0 or G0 phase. Otherwise, normally, G1 phase in labile cells should get into the next phase. The next phase is named as S phase, and this S phase is the synthesis phase. The synthesis is to refer to the important content of the dividing cell, which are the DNA. During this period, the DNA synthesis takes place. Replication of the DNA would take place. So the cell would have 46 double amount of DNA material. Initially, the DNA material of the cell was 23 paired or 2N. But at the end of S phase, you will have not 2N, but 4N. So there is now 4N level of the DNA material. And once the DNA is duplicated, it gets into the next phase. The next phase is gap 2, the second gap phase, or we call it G2 phase, where final preparation of cell division takes place, such as, for example, repairing of any damaged DNA, synthesis of tubulins for formation of microtubules, for formation of mitotic spindles are synthesized. ATP, RNA, protein required for coming mitosis are all should be in place. And the duplication of centrioles, which had begun in G1 phase, should be also completed. And again, a second checkpoint is here. This second checkpoint is to assess the proper chromosome duplication. The state of chromosome duplication, whether it is correct or not, should be assessed. And if things are all correct, again, certain proteins will be triggered, and this would lead to activation of the cell to get into the next phase. The next phase is mitosis. And during this mitosis, cells would get to divide. And this mitosis is just a brief phase, I told you, and it lead to a split of the cells into two daughter cells, which grow and take their activities in the next phase. The next phase is just nothing but G1 phase. So here, after mitosis, the cell would get into the first division of the interphase. So in labile cells, the cycle moves and moves and moves on until the organism dies. In stable cells, the cells get out of G1 phase and remain there until required. When required, it can come back. When is that time? It depends on external factors. In permanent cells, there is no more coming back to the cycle. If the cells die, they would die permanently. So the person, the organism, will lose that type of cell. No more replacements. That's why, for example, central nervous system cells are not replaceable. In case of damaged brain, you know, the healing is not possible. So it would lose them forever. But in others, the cycle moves on and on until the organism dies. So that's what I can tell you in cell cycle. And this has now brought us to finish our cytology and cell cycle. And I would ask you if you have any question to ask. Otherwise, I finish at this point. And after the break, my colleague, Dr. Salamon, would take you to another topic. So thank you very much. With this, I have finished my share, my assignment. Thank you. If you have anything to ask, feel free to ask. Okay. How the medical application of studying sex chromatin to disclose the genetic sex in patients whose external sex organ Yes, that's possible. Yes, that's possible. The presence of this X chromosome can be identified. You see, we say there are two X chromosomes in the female. Also, one is inactive, still it is there. So it can be identified. It would be identified. So that's possible. Hey, sir, is heterochromatin an active or no? Heterochromatin is inactive. It is this one. It is not active. Euchromatins are the active ones. I will give you the PowerPoint. When the PowerPoint comes, you would see it. What were neutrophils? Okay, neutrophils are white blood cells. White blood cells are 5 types. Out of the 5 types of cells, 1 type are neutrophils. This type of cells happen to have the inactive X chromosomes in the female located as an appendage. That's all. You would study about neutrophil another day. But for me, I was teaching you about the inactive X chromosomes in the female. So in this neutrophils, the inactive type of X chromosomes are appearing as a drumstick. You know, the drumstick is an appendage with a handle and the end is rounded. So it is for beating a drum. So it looks like this and they name it as a drumstick. The third checkpoint, thank you very much, you remember, it is within the mitosis. Maybe at the stage of metaphase, again, there is a checkpoint. If that checkpoint is found to be correct, mitosis would proceed. So you would hear this from next description. So I think that, okay, are labile, stable, and permanent cells divided based on, yes, you can say that, yeah. They show, I don't know what I can tell you. It's better to say that Stable and permanent cells have very much elongated G1 phase. And this part of G1 phase is named as G0. Stable cells could cross this phase, whereas permanent cells are not able to cross this G1 phase. We don't call it anymore this G1 phase, we call it G0 phase. Whereas labile cells are found in G1 phase only for a moment, unless they are defective. Unless they are defective, the checkpoint allows them to proceed into the S phase. So that's what they are. How chromatins and chromosomes are distributed in multinucleated cells, such as osteoclasts. Just same as these multinucleated cells are same as other cells which did not take cytokinesis. You know, during cell division, there is a karyokinesis, division of nucleus equally, and cytokinesis, division of the protoplasm equally. In multinucleated cells, the division of protoplasm does not take place. Otherwise, the nucleus is just, the nuclei are same as multiple cells. Yes, in many of the cases, they do have cell. Maybe if there are some exception to this, I don't know. Maybe you search to get such exceptional ones. Otherwise, they are similar. In some of the cases, like for example, in the osteoclasts, indeed, there is a good example you gave. Osteoclasts are formed by fusion of multiple cells. The cells have fused, merging their cytoplasm, whereas the nuclei remained unfused. So it is the nuclei of two cells were similar, the amount of DNA were identical, they fuse, so the multinucleated osteoclasts should have to have same, the nuclei should be similar. In some cases, the multinucleation could come after cell division, which did not complete it. So multinucleation can come by two manners. One is by fusion, such as